Hello, my name is Hannah, and I'm one of the 1802 lab instructors this semester. Today, we'll be going through some of the videos with the material to answer questions for your exercise 6 worksheet and to learn about the first part of animals. So here are our learning objectives. I'm not going to go through all of them, but this is important to read and look over, especially for preparing for the lab practical or maybe quiz 2. It's really important to know what you're supposed to be getting out of this and what you're expected to be able to recall. So this video will help answer questions for activity 1 on your worksheet and on your handout. So we'll be looking at phylum Porifera, which is sponges, and this is within the kingdom Animalia. So watch this video, read your handout, and you should be able to answer questions 1 through 4 on your worksheet. So a little bit about kingdom Animalia. These are going to be our eukaryotic, made up of eukaryotic cells, multicellular organisms. So they're going to lack cell walls. So we're going to have flexible animal cells. They're all going to be heterotrophic, which just means that they cannot self-produce their own food like plants through photosynthesis. So they need an outside source. Most animals are mobile at some point, if not most of their lives, and they follow the gametic life cycle. And we'll take a look at that in just a second. So super diverse. We're animals, so we follow this life cycle, and there's tons of ecological roles and niches throughout this kingdom. So here we're taking a look at the gametic life cycle. So we're spending most of this time as diploid organisms. So if we look here, we have humans as an example because they're most relatable. So we have 2 diploid humans, and the gametes are going to be made within the ovary and the testes. And through meiosis, they produce haploid sperm and egg cells or gametes. And through fertilization, 2 gametes come together, form a diploid zygote. And through mitosis, we get that development of the adult organism that can then again form those haploid gametes. Here is basically a taxonomy cheat sheet. So it lays out all the taxonomy that we're going to go through. And that's going to be involved with Animals 1. In this specific video, we'll be talking about Parazoa. So that's going to be no true tissues, and the phylum that's within that clade is going to be Porifera, and those are going to be our sponges. So here we're looking at our parazoans. So this only includes sponges, which is the phylum Porifera, and something that is extremely distinctive is that there's no true tissues. So they have no tissue specialization. And that's one of the main ways that these groups are divided and categorized. So one is through divided by tissues, and the other is the method of embryonic development. So we're going to be talking about the parazoans, but there is another group called eumetazoans. So that includes all other phyla, and that's going to be cells that are arranged into at least 2 tissue layers. So yeah, that would be all the rest of this. And here we're specifically looking at Parazoa, and so you can see that there's just one group or one phylum. And sponges are going to be our most primitive group because they have no tissue layers. So here, a little bit more. So the phylum Porifera, and then the clade Parazoa, so saying that we lack specialized tissues. So these are going to be asymmetrical, and I know this picture kind of throws you off a little bit, but this is asymmetrical, and that's just another distinctive morphological feature. So these sponges, they are filter feeders, so they bring in water through their pores and then expel that water through the opening in the top called an osculum. And from there, they can capture their food like nutrients and microorganisms through specialized cells called choanocytes. And these choanocytes are going to be located on the inner layer of the mesohyl. And we'll take a look at this a little bit later, but this is just a really good introduction. And then it's sandwiched between two thin layers of cells, making up the epidermis. And they again have food capture through those pores and create kind of like a current with the water, and that's how they obtain their food and are able to maintain the nutrients they need to survive. So the adult forms can't move, or they're sessile, but they do produce flagellated larvae, which can swim and find a new place to hang out and grow and reproduce. So here we're taking a look at the gametic meiosis life cycle in sponges. So sponges are hermaphrodites, so that means that they produce sperm and egg within the same sponge. And another cool thing is that they have synchronous spawning. So all of the sponges simultaneously release their sperm into the water. And again, they're always pumping and filtering that water. So they're actually bringing in the sperm, and once the sperm reaches the mesohyl, the egg is able to be formed, and that zygote, it reaches the egg that's formed, and that zygote is able to form through the merging of the sperm and the egg. So that mature larvae is then released through the top of the osculum. So yeah, it's a pretty cool process, and it's cool how they use that, kind of that natural feeding mechanism as also doubling for fertilization in a way to, maintain genetic diversity. So this slide here talks a little bit about the colonial choanoflagellate theory, and that theory is just that sponges are originally derived from choanoflagellates. So those are a free-living unicellular protist or microorganism. And here you can kind of see that they have a similar flagellated larvae-like form, and they're able to form colonies, and they have the same feeding mechanism. So that's thought to, be evolutionary. Our sponges are thought to be evolutionarily diverged from these choanoflagellates. And again, here we're looking at how they're choanoflagellates and sponges are almost identical in structure and function. So they have the ability to form colonies and pool in water the same way. And the cells are structurally similar. And we can see that here, how they're going to be pulling those food particles through the water current and able to maintain and grow through that. So here we're looking at another sponge. So here we're talking about the sponge anatomy. So we have this inner layer called the mesohyl, and then on this inner layer next to the mesohyl, we have these choanocyte feeding cells. And so these food particles are being brought into the sponge through the water current that it's created through that filter feeding mechanism. And they're pulling these through these outer pores along here. So these pink little pores with the dots. So when that current's created, the food particles are able to be absorbed by the sponge, and then the rest of the water is going to be released through the top of the osculum. So we're just talking a little bit more about that filter feeding mechanism. So here, some sponge anatomy. The choanocyte lines that inner layer of the spongocyst. So the spongocyst makes up the inner layer, and then we have our choanocytes, and then this pink middle layer between the epidermis and the choanocytes is going to be our mesohyl. And that osculum, again, that top opening part where the water is being released after it's been, after the nutrients have been absorbed. And then here is a zoomed in kind of picture of what those choanocyte cells look like. So you can see how they're really, really similar to those choanoflagellate structures. And so they have a flagella and a collar, and through that, you know, kind of that wagging motion, that's how they're able to create that water current. So we have 3 classes of sponges that are within this group that we'll be kind of talking about. And so we have class Demospongiae. So that's going to be our classic sponges, the ones we can go out to the store and buy, used to wash our dishes, used as a loofah, that sort of thing. And they're super soft, and they have cilia spicules, and they have just really soft, fibrous structural cells. So here we have class Calcera, and it kind of sounds like what it's made up of, calcium carbonate. And these form like kind of a hardened skeleton. So they're going to be, you wouldn't want to wash your body with these. It would hurt, and they're hard and calcified. And then we have class Hexadelena. So these are going to be our glass sponges, and they're composed of these cilia spicules, and they're usually created within these 6 rays. So we're seeing a really kind of advanced structure here, and that's to support an otherwise, what would be an otherwise pretty fragile makeup. So those are our 3 classes. And take a look over the handout, and hopefully you've already watched this video, and take a look at activity 1, and you should be able to answer questions 1 through 4 on your worksheet. Thank you.